Today we're going to discuss about Human Genome Project in Molecular Genetics, Unit 2, Chapter 5 in Zoology. The Human Genome Project is precisely called as HGP. The Human Genome Project was launched in the year 1990. This project was an international project and it is a mega project too. And the time of completion what they estimated in 1990 is 13-year project as 13-year project. So it took to complete as they estimated to complete this project by 13 years. That means 2003. And the human genome is 25 times larger than the genome of any organism sequenced to the date and is the first vertebrate genome to be completed. That is our human genome, that is our genome is 25 times larger than any other organism genome. And this is the first genome to be projected to study. And the first vertebrate genome to complete also. Approximately the human genome has 3 into 10 to the power of 9 base pairs. So they had estimated it 3 into 10 to the power of 9 base pairs. This they may ask in 1 mark. And why it took 13 years? Because during 1990 there is no well developed technologies. So due to that it took 13 years. And while that time along with this project the new branch was arise in this biology department, that is called bioinformatics, and it is associated with the computer and the biology. So the informations are stored in the form of database. So that has developed and a new branch was arise that is known as bioinformatics. So first you must remember the launch period of HGP. They may ask in 1 mark. 1990. It is a mega project. It took 13 years to complete. And how many base pairs are there? Approximately 3 into 10 to the power of 9 base pairs. And it is 25 times larger than any other genome. So goals of Human Genome Project. So what is the goal? That is, for what purpose they are doing this project? The first, identify all the genes in the human DNA. So they thought of there is only approximately 30,000 genes will be there. So they can easily identify the genes. So they had a thought to identify all the genes in the human genome, that is in the human DNA. That is all genes should be identified. This is the first goal. The second goal is determine the sequence of 3 billion chemical base pairs that make up the human DNA. So the human DNA is made up of 3 billion base pairs and it is sequenced that we can able to assumed or determined. Okay, so for making up a DNA, 3 billion chemical base pairs are sequenced. That we know that for that purpose. That is one of the goal to study the Human Genome Project. To store this information in database. So what are the results are coming in the project, that has to be stored in the software as databases. So to study. So this was done in 1990 and now we can check the project when we move on to the computer. When we see in the computer, we just click the Human Genome Project, we get the so many details about this project as well as what are the genes they have identified and how it is sequenced. Okay, so for that the databases are formed. And improved tools for data analysis. So the tools have to improve. We have to find, that is the new branches arise now, bioinformatics. So for that the C++, Java, like that, so many languages are also used in this for data analysis and to store the information in the database. Transfer related technologies to other sectors such as industries. By using this Human Genome Project, we can transfer the technologies. So that technologies to other sectors like industries, pharmaceutical industries. So it is used in pharmaceutical industry, the main industry, and biotechnological industries. There and all they can transfer the sectors, and addresses the ethical, legal and social issues, that is ELSI, that may arise from the project. So due to this project there is an ethical issue. Ethical means traditional issue or classical issue may arise. Legal as per the law, and the social, the society. So these issues will be arising from the project. Okay, that we have to address it. No, it doesn't give any problem to anyone, and it was secured, like that you have to give assurance. So these are the goals, you have to keep it in mind for human genome project. The first is all the genes has to be identified. Second one is to make up the DNA, you need the 3 million chemical base pairs. Okay, and the third one is information in database to store. And the fourth one is data analysis. For data analysis you need more improved tools. And transfer sectors, the technology to be transferred to the sectors like industries. And you have to address ELSI issues of this project. These are the goals. So this is a human genome project. From a man, the DNA is collected. So after collecting, so the DNA, the DNA has to be cloned with many copies. After this DNA is collected and it has to be cloned, that is, it has to be prepare more copies. Okay, this one. And after preparing the copies, you have to make it in pieces with the help of restriction endonucleases, the enzyme which cut the DNA into fragments. Okay, so the sequence are arranged in correct order. So the sequence has to be arranged in correct order, and after arranging in correct order, the complete genome has to be assembled in right order. So the assemblage of the genome in a right order. This is the normal human genome project work, and this is a shotgun whole genomic sequence. Now it is familiar and it is easy, one. So methodologies of human genome project. There are two major approaches. The first approach is identifying all the genes that are expressed as RNA. So identifying all the genes that are expressed as RNA, ESTS, that is expressed sequence tags. So all the genes have been identified as RNAs. ESTS means expressed sequence tags. This is one approach. And the second approach is sequence annotation. Sequence annotation, the sequence, you are sequencing, no? Sequencing and analyzing. Annotation is analyzing. So these are the two approaches. So you are getting the whole genome for sequencing. So a human genome means the entire, in all the cells the DNA is there, no? That complete set you are taking and you are analyzing. For what? For sequencing. So sequencing the whole set of genome was taken. And there are some genes, they have coding and non-coding sequence, and later assigning different regions in the sequence with function. So coding means they are involving, as we have studied about the protein synthesis, transcription, translation process, okay, and replication process, okay, these and all comes under coding sequence. So they are useful in preparing protein synthesis, or they are used in replication process. And non-coding sequence are there. They are not code for any genes, that is code for any replication process, but they have to be present for other purposes. So they are called non-coding sequence, and they are assigning in different regions. So they are present in different regions with functions to sequence, to arrange with functions they are present. So coding is one in one side and non-coding is one side, but they are doing their function in a sequencing order. For sequencing, the total DNA from a cell is isolated. So if you want to sequence a DNA, first you have to take the whole cell. From the whole cell, the whole DNA you have to take it out and kept in a separate slide or separate watch glass, anything. Okay. So for sequencing, the total DNA from a cell you have to be separate from it. So from the cell you have to totally separate the DNA and kept in an isolated vessel or a watch glass and convert into random fragments. So the isolated DNA you have to cut into small pieces with the help of restriction endonucleases, that is the cut enzyme. After that, the cloned, that is you have to clone it also, the DNA also. For what? For amplification of DNA. Amplification or amplified is to get large number of DNA which is used for sequencing process. Okay, sequencing process. For this, that is for cloning process, you need a suitable host and a specialized vector. Here, the bacteria and yeast are the host. And the vectors are BAC, bacterial artificial chromosome, and yeast artificial chromosome YAC are used. So, bacteria and yeast are the two hosts used in Human Genome Project, and the vectors are BAC, that is bacterial artificial chromosome, YAC, yeast artificial chromosome. Keep it in mind, they will ask in your NEET exam and in your board exam these 2 points. So now the frequency, that is the fragments. Now the fragments are sequenced using automated DNA sequencers, a machine which was developed by Frederick Sanger. So Sanger sequencer also we can say. So as we have studied here, no, in this that is the fragments for sequencing this line. So the fragments are made and they have to be arranged with the help of a machine that is called automated DNA sequencer. And this automated DNA sequencer was developed by Frederick Sanger, and this is also called as Sanger's method of sequencer, that is DNA sequencer, or Sanger sequencer also we can say it. So keep it in mind, for DNA the fragments are sequenced by the DNA sequencer which was developed by Frederick Sanger. This they may ask in 1 mark in your NEET exam also. The sequence are then arranged based on what basis the sequence is arranged. Few overlapping regions is also there, and using specialized computer based on the programs. So while arranging the sequence, one is the overlapping regions. So there are some overlapping regions, and another one is the specialized computer. They are producing the programs, no, the computer programs. On that base also they can able to sequence. So 2 methods, overlapping regions and specialized computer based programs. Okay, these 2 methods are used for sequencing. And these sequences are analyzed. So they are analyzed, annotated means analyzed, and assigned to the each chromosome. So the sequence are analyzed and assigned to the each chromosomes. They are giving the each chromosome a report on analyzing the sequence. The genetic and the physical maps on the genome are assigned using the information of polymorphism of restriction, endonucleases, recognition sites, and some repetitive DNA sequence are called microsatellite. So microsatellite they may ask in 2 mark, okay, and they may ask in your NEET and board exam as 1 mark also. So the genetic and the physical mapping, that is of the genome, on the genome, the genetic and the physical map is assigned for what purpose? Getting the information in a polymorphism in many forms. That is, polymorphism means they have many phases of DNA, many different shapes. Okay, the polymorph, here we are talking about the enzyme restriction endonucleases have many phases. So they are doing many functions, many shapes will be have, many functions will be have. So based on the polymorphism of restriction endonucleases, they have the recognition sites where the DNA has to cut into fragments. Okay, and they have to sequence, and they have the repetitive DNA sequence also. That is called microsatellite. And now the latest methods are used, that is sequencing even longer fragments. For longer fragment sequence, they are using the shotgun sequencing that we have seen in this picture, no? So this one is a shotgun sequencing method. Here we can use longer DNA fragment also in shotgun sequencing. It is the latest method, and for this we can use supercomputer also. So the traditional sequencing method are nowadays the traditional. What is the traditional means? We have seen here, no, this two methods, two approaches. So these and all the two approaches, this one and this one, this two approach methods and all, traditional one of sequencing the genome. So nowadays it was replaced by shotgun sequencing method. Okay, and it is easy to do, and it won't take as much as time. That is the 13 years project, no, like that. Too much of time it won't take nowadays because the technology is developed. So the shotgun sequencing had replaced the traditional sequencing methods. Now we move on to the salient features of Human Genome Project. So Human Genome Project contains 3 billion nucleotide bases. How many? It contains base pairs means 3 billion base pairs. They may ask in 1 mark. 3 billion nucleotide base pairs. An average gene consists of 3000 bases, the largest known human gene being dystrophin with 2.4 million base pairs. That is, in human gene there are how many nucleotide bases? 3 billion nucleotide bases. And the average gene consists, that is, the averagely the gene has 3000 bases. And the largest of human gene we know is dystrophin. So dystrophin is a protein which code for muscle strengthening. And there is a dystrophy also because the muscle, this gene doesn't work, it doesn't do its function. It causes muscle dystrophy. Due to that, the muscle problem will arise. Okay. So the dystrophin gene has 2.4 million bases. So they may ask this as also in your 1 mark. The genes are distributed over 24 chromosomes. How they are distributed? 24 chromosomes, and that is 19th chromosome have highest gene density. In 1 mark they may ask. So the 19th chromosome has highest gene density, and the 13th chromosome and Y chromosome has lowest gene density. So based on this chromosomal organization, human genes show diversity, that is differences. So we will see these points once again as a recapitulation. So human gene has 3 billion nucleotide bases. So average the gene has 3000 bases, and the largest human gene is dystrophin, which code for muscle protein. If it has any problem in the muscle due to this dystrophin, then that disease abnormality is called as dystrophy. And this dystrophin has 2.4 million bases. and the genes are distributed over 24 chromosomes. They are distributed, and the 19th chromosome has highest gene density, and the 13th chromosome and Y chromosome have lowest gene density. Due to this organization, the human genes have diversity, that is differences in their density. There may be 35,000 to 40,000 genes in the genome, and almost 99.9 nucleotide bases are exactly the same to all the people. That is, this is an approximate one only. 35,040 genes are present in the Human Genome Project, and 99.9 nucleotide bases are same to all the people in the world. They say it according to their project. The functions for over 50% of the discovered genes are unknown. So 50% of genes, they don't know what they are doing, their functions. So, but there are only, they had find out only the 50% of Human Genome Project, and the remaining 15% they don't know what is the function of the genes, but they are there in the genome. Less than 2% of gene codes for proteins. So for proteins, how that is, in 100%, only 2% are code for proteins in a Human Genome Project. Repeated sequence make up very large portion of the human genome. So why it becomes too large means the repeated sequences are more. Due to the repetition, the human genome becomes larger. So the repetitive sequence have no direct coding the functions, but they shed light on the chromosome structure, dynamics and evolution. So the repeated sequence, they don't have any direct function. They doesn't code for anything. But they have a role in the chromosome structure, evolution and dynamics. That is, they shows the genetic diversity, how the evolution have evolved in from one organism to another organism, and how they play the role in the world, how they evolved and what are the characteristics. So they won't code for that is non-coding. As I talking about the non-coding sequence in the methods, the non-coding sequence have more repetitive sequence which are not functional, but they have the characteristics to light on the evolution and to play a genetic diversity role. Genetically they have to form the different for this, okay, form the different organisms, okay. And the chromosome 1 has 2968 genes, whereas chromosome Y has 231 genes. Keep it in mind, they may ask in your board exam and in your NEET exam as 1 mark. So now we will see these points recapitulation. So 35,000 to 40,000 genes in a genome, and 99.9 nucleotide bases are same for all people, and 50% of the gene function is not discovered till now. Less than 2% is code for proteins in the genome. Repeated sequence occupies the larger portion of the genome, and repeated sequence doesn't have any function, but they play an important role in chromosome structure, evolution and forms the genetic diversity. The chromosome 1 has 2968 genes and chromosome Y has 231 genes. And scientists have identified about 1.4 million locations where the single base DNA differences occur in human. So the single base DNA differences, that is 1.4 million locations are identified by the scientists, and they find the difference in the single base differences. That is, the differences was seen in the human, how they are differ from one person to other person. Like that, the bases are different in the single base DNA. That how many bases they had found out, that is in the sites 1.4 million locations, and they are known as SNP, single nucleotide polymorphism. SNPs means single nucleotide polymorphism, and you can pronounce it as SNIPS, SNPs also. So keep it in mind, the scientists have found out the location, 1.4 million location of single base DNA shows the difference in the human being. And S and P, single nucleotide polymorphism, and you can pronounce it as SNPs. So identification of SNPs is helpful in finding the chromosomal location for the disease associated sequences and tracing the human history. So SNPs is used for finding the chromosomal locations, that is this one. What the SNPs, what is the single base? So it finds the disease, genetic disease in the human history. The SNPs is used. And as we have discussed, this Human Genome Project is a mega project with 13 years as they estimated, but they didn't complete it by 13 years, that is 2003 they didn't complete it. They taken extra 3 years, that is they completed by 2006. Why? They had complete almost all the genes, but the chromosome 1 has more genes that we have discussed now. 2009, that is 2009 68. 2009 68 genes are seen in chromosome 1. Because of this only, they taken extra 3 years to complete. So this may ask in your NEET exam, how many years to complete? Estimated year is 13 years, but the completed year is 16 years. That is 2003 is the estimated year, but it extends till 2006. Applications and future challenges. The mapping of human chromosomes is possible to examine a person's DNA and to identify the genetic abnormalities. So, genetic abnormalities and person DNA is easily to examine it. This is extremely useful in diagnosing disease and to provide genetic counseling to those planning to have children. So if they have any problem and that genetic disease have been transferred to their younger other generation, so that was found out by the doctors, and they will give the genetic counseling with the help of this HGP project, and it is a useful one because we can create a normal society with the help of Human Genome Project. But it was against the nature, but it is good. So we cannot. So it is good to the society to not giving the genetic disabled person. So this kind of information would also create possibility for new gene therapies. So new gene therapies has to be evolved with the help of Genome Project. Besides providing clues to understand the human biology, learning about non-human organisms. So we can able to understand human biology instead of going to the non-human organism. For example, usually a medicine or a vaccine is coming. What they will do? They will go for an rat examination or rabbit examination. So if you know the molecular structure of a human being, you cannot go for rabbit examination or rat examination first. Directly you can go for the human being itself if you know the genome. That only this point is telling. DNA sequence can lead to an understanding of their natural capabilities that can be applied towards solving challenges. So that is, DNA sequence is helpful to understand the natural capabilities and solving the challenges which arises now. Healthcare, agriculture, energy production, environmental remediation. So healthcare, disease-free world can be formed. Agriculture, new varieties with small season period of harvesting, minimum period of harvesting. That type of agriculture can be used. Energy production like electricity, water, wind, these and our energy. So this is also capable of using this. Environmental remediation, that is bioremediation, that is the environmental can be free from pollution by using this. These are the challenges. By DNA sequencing, we can come across these challenges and we can improve these fields too. A new era of molecular medicine characterized by looking into the most fundamental causes of disease than treating the symptoms will be an important advantages. So molecular medicine based on the DNA which the disease was caused in the human, that based on that you can create a medicine and you can give it and you can rectify the problem by seeing the symptoms well in advance when you know the DNA of the organism which causes disease in the human being and where it attacks. So that can be easily done by this human genome project. Once the genetic sequence become easier to determine, some people may attempt to use this information for profit or the political power. This is a disadvantage. Okay, disadvantage of the human genome project. So if there is a good, there is a bad. So if the information was given for the profit to get the power, political power, they can able to misuse also. And the insurance companies may refuse to insure people at genetic risk. So if they have at a genetic risk, so they won't insure to the particular persons. Okay. So the future medical bill incurred is less than the perfect people. So the perfect people doesn't have any problem, means they won't go for insurance, medical insurance. So like that. So less than a perfect people. Another fear is that attempts are being made to breed out certain genes of people from the human population in order to create a perfect race. So that is genetic abnormalities is not there in the society, or a disabled person will not be in the society, that is eugenics. The field of eugenics and euthanics can be created here with the help of this, and a perfect race. Only the healthy and abled persons will be there in the world, and the disabled persons will not be produced at all. If also produced, they will kill them. So like that also can be attempt in future. It is a big risk to the human race and against the nature also. I think you have understand the human genome project, the goals and the methodologies, salient features and the applications and the challenges to overcome. And it is a very important question based on the salient features of human genome project. It is an important question, and the goals is also an important question. They may ask, the applications is also an important question. So they may ask in your board exam if you understand. Okay, I think if you are understand it. And now this you must know. The pharmacogenomics is the study of how genes affect a person's response to drugs. This relatively new field, this is a new field, combined pharmacology, the science of drugs and genomics, the study of genes and their functions to develop effective, safe medication and doses that will be tailored to a person on genetic makeup. Based on the genetic makeup, the drugs, that is the medicines, will be produced, and it is a new field, pharmacogenomics, genomics field. Hope you understand the topic. If you have any doubts, post your doubts in the comment box. Give a thumbs up, share and subscribe to Science Easy Tech channel.